Hello, my name's Matthew. I just graduated from Duke University last weekend. I majored in biology, and my advisor is Thomas Schultz with the Nicholas School of the Environment. And so my research is on how Superfund site pollution remediation efforts are driving genome changes in pollution-resistant killifish. So to give a little bit of background, my project is situated in the Elizabeth River watershed, which is a very important economic area to the United States. Over 500,000 people live and work near the watershed, and it's nearby major cities such as like Portsmouth, Virginia, and like Norfolk, and it's home to lots of industries and even the largest naval base in the United States. However, one potential problem is like in the Elizabeth River, in the past, there's been centuries of wood treatment plants releasing lots of polycyclic aromatic hydrocarbons into the sediment, and these PAHs. Or PAHs for short. And we know that these pose a lot of significant threats and potential environmental and human health hazards because they are known to increase your risk of cancer, suppress your immune system, and even increase the risk of cardiovascular disease development. So this is very important. And so the main potential site that we're actually looking at in the Elizabeth River watershed is called Atlantic Wood Industries Superfund site. And if you don't know what a Superfund site is, that's basically just a heavily polluted area that's been marked off by the EPA for extensive pollution cleanup operations. And so, however, what's really remarkable is that there's actually Atlantic killifish populations that have actually lived at the Superfund site for an extensive period of time, and they've actually managed to evolve resistance to these nasty chemicals. And they achieve this through a variety of adaptations. For example, they have variants in genes that enable them to better process and metabolize these cancer-causing compounds. And then they also have increased antioxidant pathways and variants of genes important in implications of cancer. So that also shows why they're also a really cool potential human health model organism with lots of cool human health applications. And yeah, so however, what we do know is that these adaptations to these pollutants don't come free. There's a lot of trade-offs. For example, killifish that live at Atlantic woods, even though they're able to resist these pollutants, on the other hand, they're a lot more sluggish than fish at less polluted sites, and which it basically makes them more vulnerable to predators. So we do know, and there's also a really high metabolism cost of having these adaptations. So basically just a lot of trade-offs that come along with this resistance. And so basically that brings me to the crux of my research question. And so lots of previous literature has already established that these killifish populations have been being driven far apart and variant due to pollution. Pollution is driving populations farther away from clean sites. However, what's really interesting, however, is that in 2017, the EPA actually completed the largest phase of the cleanup operations at the Atlantic Wood Industries Superfund site. So basically what my project is looking at is, so we know that previously pollution has been driving these killifish populations to become more variant from each other. Now that this heavily polluted site has been remediated and cleaned up, how is that impacting the genomes of these killifish? So basically, my research questions are, how has anthropogenic pollution remediation driving the killifish genome of previously resistant killifish, and what specific regions of the genome are actually under selection? So for my materials and methods, well, we had a lot of data from killifish from various populations before the remediation effort, and then after the remediation effort, we collected a lot of samples from killifish and then sequenced them, mapped them to the reference genome, and then basically just called variants. And then basically what we did was we compared this post-remediation data to pre-remediation data to see how the population structure has been changing in response to these remediation efforts. And for that we used DAPC and PCAdapt, which are both principal component analysis-based methods. So for the first results, if you look before remediation, Atlantic woods killifish cluster very distinctly and far away from Kings Creek, which is the clean reference site, and Jones, which is a moderately polluted site. However, after post-remediation, and these killifish have been living for a few years without the pressure of environmental pollutants, what we see is that now Kings Creek is still far away, Republic is still far away, but now what we see is the program is no longer able to genetically distinguish Jones killifish from Atlantic woods killifish. And this is really interesting because what this is showing is how before remediation, Atlantic woods being a super heavily polluted population is like very variant from everything else. But now, after the remediation efforts have like cleaned up a lot of the pollution, now Atlantic woods is so genetically similar to Jones, which are moderately polluted fish, that DAPC is no longer able to genetically distinguish these two populations. And so what we basically think this is showing is that now Atlantic woods killifish are now very genetically similar and probably express like the phenotypes of a moderately polluted population rather than a heavily polluted population. Alright, so for like the second part of our results, we were basically looking at the specific outliers that could be explaining these differences between your post-remediation and pre-remediation population. And we used principal component analysis and just found 118 unique single nucleotide polymorphisms and looked at candidate genes, 100,000 base pairs upstream and downstream that could be explaining that could be responsible for these SNPs. So basically, in summary, what was really cool about this project is we found how the population genetics of killifish at the Atlantic Woods Industry Superfund site have indeed shifted dramatically. Before remediation, Atlantic Woods killifish were very far away. However, after remediation, they have now shifted to become genetically almost identical to the Jones, which is moderately polluted, and this is just showing how after remediation, genetically these killifish have moved from looking like fish that are really pollution resistant to only moderately pollution resistant.